All right. So let's start with what we have discussed in the previous class. Quickly, within 5 minutes, we can have a quick recap. So clinical trials is the one which we started with, because before we go with the clinical SAS programming, we want to understand the domain first. So we started with the clinical trials. So clinical trials, there are certain phases, right? So why we do clinical trials, irrespective of whether it is a treatment drug or it is going to be a vaccine, that is nothing but a preventive drug, or it is going to be a medical device, any medical device. Before it come to the market, it need to undergo some process. That process is nothing but a clinical trials. Why do we do this? We are doing this to ensure the safety and efficacy. These two are the two main reasons why we are going to the clinical trials. First and foremost thing is going to be, I need to ensure that whatever the device or the vaccine or the treatment, the drug that we are taking, that's supposed to be safe enough, that's supposed to be efficient. So to understand these two, primarily we go with the clinical trials. In addition to that, there are some other factors as well. Those factors are going to be the dosage information, how frequently we need to take this particular one, what is the mechanism, right? So what are the side effects? All these are going to be the sub factors, but the primary factors are going to be safety and efficacy. Clinical trials initially starts with preclinical trials. In a simple way, if I want to talk, preclinical trials is primarily done on or conduct on animals. Once we understand that this is risk-free, then we actually enter into the clinical trials. In the clinical trials, there are 4 phases. Phase number 1, it generally carry out on human beings only, and that too, we are going to conduct this only on the healthy people, right? Again, last week I haven't informed one thing. We consider healthy people not in all the cases. There are some exclusions here also. Consider there are cases like HIV, right? On a HIV, you cannot conduct your HIV trials on a healthy people. Here, I need to conduct this only on the normal people, only on the people who are already suffering with this particular disease. Guys, am I audible? Can someone please confirm?
Yes.
Okay, Santosh, I think, because I got a message from Santosh saying that he is not able to hear me. That's the reason I asked you. Okay, thank you for the confirmation. Just give me a second. Okay, done. Right. So consider the cases like HIV, cancer, and all. In these cases, I cannot conduct even the phase 1 trial on the healthy people. Instead, we will do it only on the people who are already suffering with the disease. It is generally carried out with a group of 10 to 30 people. Sometimes it can extend even to the 80 people. The primary reason why do we do this is going to be the safety. Safety is the primary reason, our primary outcome that we are expecting here. Then we have phase 2. What we are going to do under the phase 2, we will extend our group from 100 to 500 people. Here we are going to have the mix of the people. That is nothing but we go with the healthy people. We also take care of some patients. While considering the patients, yeah, we consider the patients who are suffering with the same disease, and we also consider the patient who are suffering with the different diseases. Correct? So same disease as well as the other disease. Both the patients we are going to consider. At the same time, we are also going to consider the healthy subjects. Here, the primary reason, the primary thing that I am expecting is going to be efficacy. Then we enter into the phase 3. In the phase 3, the most important phase is always the phase 3. Here, we extend our base to 1,000 to 5,000 people. Here, we are going to consider all the ranges of the people, different groups, different genders, different age groups, different continents, healthy subjects, people suffering with the same disease, other diseases, whatnot. We are going to consider a lot of things here. And again, the primary reason of doing this is going to be to meet both of them, safety as well as efficacy, At the same time, along with these two, we also discuss about other factors which are nothing but the side effects, dosage, how frequently you need to take this. All these information we are going to do here. Then we go into the phase 4. Phase 4 is nothing but one step before launching this particular drug. Here we are going to test this on minimum of 10,000 people to the 50,000 people, and also at this particular stage we do the marketing, we decide the price of the particular drug, all the things, marketing strategies, stock, every piece of information we are going to maintain. That's going to be the end of your clinical trials. Once everything is approved, then we are going to prepare the reports, and these reports we are going to send it for the approval. Generally, we'll send this approval to the regulatory bodies. For USA, it is going to be FDA. For Europe, it is EMA. And when it comes to the India, it is going to be our drug control. Correct? So for the respective countries, we have the different regulatory bodies. To the regulatory bodies, we are going to send this. This is a simple piece of the clinical trials. And all this we have already discussed in the previous class. Correct? So what we do today, in every clinical trial, very important. So where it will start, we are going to discuss today. Okay? Just give me a moment. Hope all of you are able to see my screen. So I'm just going ahead with a very simple introduction part we have already done. So I don't go with this particular one. Phase 1, phase 2, phase 3, all the things are there. I don't go with this particular one. So here, understand this particular piece, right? So what is the difference between a normal SAS programmer and a clinical SAS programmer? Can someone please differentiate this? Is there any difference between these two guys? What do you think? Anyone? Clinical SAS programmer apply their skills on the clinical research. Maybe we can extend the same to others as well. I can say that when it comes to banking professional, he or she will apply the clinical SAS skills to the SAS skill to the banking, SAS skill to the healthcare, SAS skills to the logistics. One way what you said is absolutely correct, no doubt about it. No doubt about it. Maybe that we can fine tune a little. The simple difference is going to be, if you are a normal SAS programmer, let's say that I'm just working for one of the bank, let's say ABC Bank, right? Without knowing the banking terminology, let's say that I have something called secured loan. I don't know what is secured loan is. I don't know what is unsecured loan is. What do you mean by mortgage? I don't know, right? So what is called deposits? What are the various deposits are there, right? What is the difference between general insurance and life insurance? Or what is called Vickers, or what is called Silas, right? Or how your QR is going to work, or how your APIs are work, or how your PVDs works, right? How various marketplaces work. I don't know any of this. What is the usage of Adobe Analytics? Adobe Analytics. I don't know all this, but without knowing all these things, the moment I have the data set with me, or the moment I have the data with me, and when I have the problem statement, I can provide the meaningful solutions for that. I can do that. Because even without knowing this information, we can still work on banking industry. Of course, banking industry, again, if you go to something like AML, anti-money laundering, right? They definitely require the banking knowledge. Without knowing what the anti-money laundering schemas and all, you cannot work on it. Without knowing the KYC, you cannot work on the KYC. But other than these two, 90% of the cases, I can happily work on any of the bank product without having any domain knowledge. Right? Same goes to your healthcare also, right? As long as I have where the claims are coming from, what are the claims, and what is the business logic. If someone shares the business logic, I can work on that. I can provide the fraudulent claims to you, right? But when it comes to the clinical SAS programmer, without knowing the clinical standards, without knowing clinical domain, you cannot work on it. As simple as that, right? A clinical domain, the knowledge is very, very important. Without that, you cannot do, right? So you understand here, a clinical SAS programmer requires to understand the objectives of the clinical trial study. What is a clinical trial study? The objectives, the phases, the study design, all these things we must be aware of. Without this, we cannot work at all, right? So that is the reason, because you all, most of you are, I can say that 100% of you are already completed your SAS programming. You know the ABCD of your SAS programming. But why you are here? Because you want to understand this piece, the second piece that I'm talking here. So what is the second piece here? The second piece that I'm talking here is going to be what is called here clinical trial studies. What are the clinical phases? What is the study design? Right? So that's the reason we need to understand the basics first. So these three are, along with these three, there are certain elements as well. These elements are very, very important and crucial to us. Like I said in the previous class, on our Friday class, I told you that, Thursday class, I told you that before we enter into the SDTM, ADaM, or PLF, we need to understand few of the basics. We need to understand some of the definitions. We understand some of the documents. Right? That work we have started now, right? So the first step in initiating the clinical trial is writing a protocol. What is the first thing? We need to write a clinical trial protocol, or I can say that any clinical research will start with this particular one. So it is the entry step for any clinical trials. Now the question is, only what exactly it is? A clinical trial protocol is a simple document, but you can consider that it is a Bible for clinical trials. Every clinical trial is completely carried out only based on the details that are present in the clinical protocol. Whatever is mentioned as is, you need to conduct that. No violations. In certain cases, there will be few violations, but those violations also should be within the limits, right? Some people also write in the protocol, these might be the violations you can expect as well. You are going to take the amendment for that as well, right? But on a lighter note, I can say that every clinical trial will start with the protocol. Okay, I said that this is a document. So who will prepare this document? This document will ideally prepare by sponsor. So who is sponsor? The person who is giving us the funds. The person who is actually giving the fund to this entire clinical research. Sometimes a sponsor can be an individual person. Sometimes a sponsor can be an organization as well. If it is an organization, you call it as a CRO, contract research organization. So a clinical trial protocol which will have end-to-end details about how you can carry out your clinical trial will be prepared by a sponsor or contract research organization. These are none other than the people who will provide the funding to our research. Clear? The very first thing that all of us need to understand is going to be the first step in initiating the clinical trial is writing the protocol. But if someone asks you who will write this protocol will be written either by the sponsor or by the contract research organization. Then a sponsor can directly write this protocol. Let's say that I am the sponsor, I have enough money. All right, I have some billions of money with me and I want to initiate some research on, let's say that, increasing the longevity of the human being. So let's say that the people are now living for 80 to 90 years. I want to increase the longevity. For that, I want to provide the funds to some of the bunch of the scientists. But I cannot prepare the protocol, right? I can only provide the funding. I have an idea, right? But I don't have the details about how to write it, where I can get this particular protocol sample, right? What and all the fields that should be part of this particular protocol. All these details are not with me. So I need someone help to prepare this particular protocol. Correct? Sorry. Now, who? All right. Now, who will prepare this protocol? Sponsor, but with the help of who? Right? So here you can see an investigator, or we can also call it as clinician. A clinician is clinician, or even if you consider the investigator, both of them are referring the same person, which is nothing but a doctor. Nothing but a doctor. A clinician or an investigator, along with the biostatistician, will help or will prepare this protocol. Right? You can ask me why biostatistician and why clinician, or simply why a doctor. The doctor is the one who's supposed to go with this particular entire trial. He is the one who will conduct, because he is the only one who have the deeper understanding about the disease, or who is having the deeper understanding about the drugs or even the procedures. So a doctor must be there. A doctor, we can here call it as an investigator. Right? Again, or if you say that Murli, why you have two different names? You have something called clinician and you have something called investigator. Is there a difference between them? If you strictly ask that, again, there is a difference. A clinician is the one who will take care of the patients, who will take care of the patients. Even he or she may directly or indirectly contact patients. Right? That is the deeper understanding. An investigator is the one who will be in contact with the patients at the same time. He or she are the one who is going to conduct the entire clinical trial. Entire clinical trial. Right? A clinician may not be part of your entire phase 1, phase 2, phase 3, phase 4. But an investigator is the one who will be throughout your disease. Okay? So the first thing that we need to understand is the protocol document will be prepared at the site of your CRO or the sponsor site. A sponsor alone cannot produce this particular one. He would expect some help from the either or both of your statisticians and clinicians. Once the investigator receives the protocol, first they will determine, let's say that I received a protocol. I cannot blindly start my clinical trials. I need to check the feasibility, right? Whether these clinical trials are possible or not. These are in realistic mode or not, right? If they are realistic mode, then only I can enter into the clinical trial, right? If someone asks you, how do you define the, whether the feasibility is there or not, right? So for an example, if I say that drastically I want to increase the height of a person, I need a drug to increase the height of a person by, let's say that 5 inches or 6 inches. How practically it is possible? May or may not. Check the feasibility. Check, are there any history where there is some drug which help us to increase the height of a person, right? So like that, I need to check the feasibility first. First of all, it is practically possible or not. Once we understand that, yes, on a lighter note or at a very high level, we understand that this particular clinical trial, carrying out this particular clinical trial is feasible. Right. Again, before we kickstart with our clinical study, again we ask few questions ourselves. What are they? Does the investigator have the necessary patient population? What do you mean by population here? What do you mean by population here? Number of subjects. That is nothing but number of patients or number of volunteers, healthy volunteers. Because I want to carry out this particular trial, do you have enough number of people? Let's say that your sponsor has given only 2 people to you and asked you to carry on your clinical trials phase 1. Can we do it? Practically it is impossible just with 2 subjects. We already know the boundaries, minimum of 10, maximum of 80. Sorry, minimum of 30, maximum of 80. Those are the minimum numbers I'm expecting, right? The minimum population based on the previous values are going to be 30 to 80, but I doesn't have 30 to 80 people now. You have given me just 2 people and you ask me to carry out the analysis. I cannot do that. right? So the first thing I need to understand here is, do I have enough population or not? Who will decide the population? The population will be decided by the biostatistician. Not only for this, for all the phases, correct? So for the second phase, we have around 100 to 500. For the third phase, we have 1,000 to 5,000 people. And for the fourth phase, we have 10,000 to 50,000 people. All these phases, the randomization, the random number of people, all these algorithms and the numbers will be produced by your biostatistician. Okay. Second, is the protocol well designed and clearly understood by the study team at the trial site? What do you mean by trial site? I told you all these clinical trials will carry out either at a community clinic or at a doctor's site or at a research site in a hospital, right? These are the various places where the research will happen. These are the various places where the clinical trial will conduct. Now the question is going to be, whoever at the trial site, are they clearly understand the entire protocol or not? Because the protocol is going to give all the details. Whatever the details that are mentioned in the protocol need to be clearly understood by all the people who are working at that particular trial site, right? Even though the protocol is good, but if I don't have enough people who can support it, can I take care of that particular one? Not possible, right? Not possible, right? Because I need to have a clear and crisp understanding about the complete details present in the protocol. That is the reason we are also studying the protocol now. Okay? Then, are the study timelines are reasonable? Let's say COVID. If government asks us or asks all the researchers, hey, I want the vaccine, right? But I'm giving you a timeline, that the timeline is going to be 3 months. I'm giving you the protocol, what need to be tested, everything, how the procedures are going to be, everything I'm giving to you. The protocol is crystal clear. All the people who are working at the different sites also understand the protocol. Fine. The first two things are done. I have enough people who are volunteering this particular research. So the first two questions are answered already. Now the third question is, the government saying that I want this particular stuff need to be completed in 3 months. But the timelines are not reasonable now. These are not realistic. For sure I cannot complete this particular within the 3 months of the time. So I definitely need additional time. So the timelines are not reasonable. I cannot quote the exact timelines, but I'm 200% sure that 3 months window for bringing up a new vaccine for protecting against COVID is highly impossible. So we need to understand. We need to have the proper population. We need to have the protocol need to be understood by all the people who are at the trial site, and also the final thing is going to be the timelines must be reasonable. Clear up to this? Why we are talking about the clinical trials protocol? Why it is important? Can I have a quick confirmation from everyone? Is this understandable? Yes or no? Any queries? All right. Next thing. So we only talk at a very high level. We said that there must be a protocol document. We said that who prepare it, with the help of whom it is getting prepared. Only that we have discussed. Exactly what it is, exactly what is called protocol document, and what are the various things that are present in this particular document, we need to understand. Correct? For that, we see this particular one. So what exactly this clinical trial protocol document consists of? Number one, a clinical trial protocol document. It has the details about the first and foremost thing, very, very important, how the clinical trial will be conducted. Right? You have phase 1, phase 2, phase 3, phase 4, preclinical, all this, right? How it need to be conducted? What need to be tested? Number 2, what are the objectives of this particular clinical trial? Let's say COVID vaccine. What is the objective of the COVID vaccine? We need to ensure that we need to bring a vaccine. It should be protective against the COVID. As simple as that, right? Let's consider polio. The polio vaccine objective is going to be, once the person has taken this particular vaccine, the person should not affect with the polio at all. Correct? What is the objective? There are a lot of stuff again. Primary objective, secondary objective, primary outcome, secondary outcome. Lot of stuff will be there. We will discuss that later. But on a lighter note, why it is required, what exactly it describes, how you are going to conduct your clinical trial, what are the objectives. Number 3, design. Number 6, when I say design, there are a lot of design procedures. Each design procedure we need to understand. Something like cross functional study, or parallel study, or parallel design. There are a lot of designs available. So what design you are using for this particular protocol? There are n number of methodologies that are available to us. So what methodology you are using for this particular protocol? Statistical considerations. We need to say, what are the population limits? So when I go with the results, what are the percentages that I'm looking at? It should be 100% accurate, or 80% accuracy is fine. When I go with the efficacy, what is the level of efficacy I'm looking at? What are the standards that I'm maintaining? All these will be part of your statistical considerations, right? Then, organization of the trial to guarantee the safety of the participants, because you are also giving the healthy participant to them, right? If you give the healthy participant to them, what is the level of guarantee that I'm getting that these patients will not fall sick? These patients are not going to be ceased. How I can give the safety consideration of this particular people? And last one, this is very, 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 very important. Most of us will take this in a light manner, but very important. What is the integrity of the collected data? That is, you collected the data, right? I'll tell you a very simple example. Most of you might aware of a very rare blood group called Bombay blood. You might know this. Only 10% of the population have this particular one across the world. That is called Bombay blood, right? So we are carrying out some clinical research. For the clinical research, what we did is we looked for some of the healthy volunteers. Let's say that 2 volunteers has come up with this particular trial. So we know these 2 people. We carried out our entire analysis. The clinical trial is complete. We have sent these 2 people again outside. That is nothing but trial is completed. They are free enough to walk into the normal world. But your data is leaked, either intentionally or due to some reason the data is leaked. So what do you mean by data leak here? You expose these 2 people to the external world, saying that there are 2 people who have Bombay blood group. Since it's a very rare group, so what will happen? Anyone who is in badly need of this particular blood group, right? They illegally also can contact these 2 people. If these 2 people are not willing to, donate their blood, they can go to the any extent. Correct or not? So the data leak is going to be the major problem. So you need to maintain the integrity of that particular data as well. You need to ensure that this data is highly confidential. We will not expose this data to the external world. All these measures you need to do. So for that, what are the various approaches that we are doing? All this information you need to keep in this particular document. First of all, you understand what it describes. So what it is describing, how the clinical trial conducted, what is the objective, what is the design, what is the methodology, what are the considerations, which organization is doing it, and how they are providing the safety to the participants, and finally, how they are maintaining the integrity of the data that we have collected. Is this clear? What a clinical trial protocol document consists of? Everyone, can I have a quick confirmation? Any questions here, guys? Wonderful. Next, this is extension of the first thing. What is this? Again, I'm talking about what is clinical trial protocol? Why is this clinical trial protocol needed? Maybe I can extend the same here. Why? The protocol is the key quality control tool for all the aspects of the clinical trial. Why? Because what do you mean by quality? How my clinical trial is happening? What is the outcome? Is that outcome is the correct outcome or not? Which is nothing but the quality control only, right? So since the protocol has all these parameters, if I follow these parameters as is, I can 200% ensure that our quality is going to be good or the outcome is going to be good. Correct? Again, the same thing. So what this clinical trial protocol document is going to be prescribed, or what it is going to ensure? It ensures that the health and safety of the study participants. This is what I have also mentioned here. It provides the precise study plan. Here it is. Design. It defines and manages the trial. That is nothing but methodology and design. It should be strictly followed by all the study investigators. That is, and all the doctors who are helping to carry out this particular trial. They need to strictly follow this particular one. Again, it guarantees the integrity of the data. We mentioned this here, allowing the combination and comparison of data across all the investigators and the study sites. Sometimes you may not be able to carry on the clinical trials at a single site. There will be multiple sites across the globe. At multiple sites, we are carrying the same study. In that particular case, we need to collate the data. So for that particular one also, we are doing with the clinical trials, right? So this is purely for the inquiry purposes. What is the clinical trial protocol? What it consists of? What are the elements of that? Each element we need to understand. Remember, each element we need to understand. All right. Let me go to the next one. So I just have given the information about protocol on a lighter note. Now, if someone asks you a good protocol, a good clinical trial protocol, what are the elements it should consist of? These are the elements. What is that? Title. This is very generic. Background information. What is the objective? Study design. Selection and exclusion of the subjects. Treatment of the subjects. How I am assessing the efficacy. How I am assessing the safety. What are the adverse events? Is there a discontinuation of the study? What are the statistics? What is the quality control? What are the ethics that I'm following? How I am handling the data? How I am keeping my records? Is there any publication policy? Right? What is the project timetable or flowchart? What are the references I'm considering? Finally, what are the supplements and analysis? Right? Why I am going with this is for the next 2 classes, we are going to discuss all this, everything. What is a study design? What are the types of study design we have? If someone says that what is selection or exclusion of subjects, on what basis you do the selection, on what basis you do the exclusion, right? Treatment of the subjects. What are the different types of treatments we have? How you go with the efficacy? What do you mean by events? What do you mean by adverse events? Right? Under what cases you go with the discontinuity study? What are the statistics? Everything, whatever you can see it on the screen, each and every topic that is present here, elaborately we are going to discuss. Once we complete this particular one, I can say that that is the end of theoretical stuff, which is part of your clinical trials information. Then we can happily enter into the SDTM. So to do that, or to enter into the SDTM, we may need to wait for 2 more classes. Clear? This is just for your reference purpose. Now, let me show you one of your template. This template, you can download it from the website. I'll give you the website details also. All right. So this is one of the protocol template. So generally, we take this template, we are going to fill it. So what and all we have mentioned here, we said that it should have title, page are general information. Can you see this? Well, who is the sponsor? Because the sponsor is the one who is going to prepare this particular work. Sponsor or the CRO. Correct? That is contract research organization. Now you can see protocol name. That is what I mentioned here. Title page is nothing but the protocol name. The protocol number, version date, we may have multiple versions. So what is the product, the IND number, development phase, who is the principal investigator, who is going to be the medical monitor, who is going to be the coordinating center. Remember, these also we are going to discuss. That is, who and all involved in the clinical study. What are the different, what and all the teams which will participate in the clinical study, right? So what is the work of each and every team? That also we need to discuss, right? So that is also part of your, I can say, definitions. Okay, now scroll down a little. Protocol agreement. I'll share this particular template with you guys. All of you kindly look at this. This is the one. This is a very, very important one. You can see there will be approximately 17 different topics here. All these 17 different topics are part of this. Okay, see this. One by one we go. I'll share this particular one. Later you can look at it, but still I'll just give you the few details. Background. Identify the product to be studied and describe it briefly. So what is the product or what is the drug? Is it going to be a developed treatment drug, or it is going to be a preventive drug, or it is going to be a medical device? Give the information of that, right? So if this particular product have some non-clinical information, what is the significance of it? Give the summary of it. So for clinical studies, give the clinical data, because everyone requires the data, right? So give the data. So here you can see that the risk or benefit section, and you can also see study objectives. What is the primary objective? What is going to be the secondary objective? What is the study design? Can you see this particular one? This is a single center, double-blinded, placebo-controlled, randomized, incomplete block, 3-period crossover trial. Can you see this? This is the study design. We need to know all this information. That is what is called single center, what is called double-blinded, what is called placebo-controlled, how randomization is happening, what is called incomplete block, what is called 3-period crossover trial. So each one of this we need to understand. Here it is only double-blinded. Here it is going to be placebo-controlled. What is called placebo, right? What is called 3-period crossover trial. These are the definitions, because these are the only interview questions. When you say clinical trials, these are the clinical trials questions, right? So we need to understand this particular one. So what is primary efficacy endpoints, secondary efficacy endpoints, how you are actually defining your endpoints, right? So this is a template. Whatever you can see here is a template. There is another thing as well. Let me just give me a moment. Just allow me a second. I'm trying to. Here it is one. This is one of the protocol. This is defined by WHO. This is defined by WHO. Yeah, World Health Organization. Here also you are going to have the same thing. More or less you are going to have the same thing. Here the template is slightly different. Slightly different. But on a whole, more or less it is going to be the same. Right? Here you can see block randomization with randomly varying block size. Right? Here also blinding or masking is going to be, the blinding is going to be placebo. Right? And it is going to be a saline one and it is a oral one. So what kind of blinding it is going to be? How I am collecting the samples? Every piece of information must be entered. So there also we talked about the adverse events, right? Here you can see the adverse events. The adverse events is going to be health status of participants will be monitored and reported as a study outcomes, right? So at this particular point of time, you may feel that This looks little difficult to me, maybe at this particular point of time, right? But once we understand all these topics, that is nothing but what is called placebo, randomization, and what is called adverse events, what are the treatment methods. Once we understand all these parameters, and when you start reading this particular document, you don't find difficult at all. And even I can say that you feel comfortable with this because you know how to read this now, right? Till that you don't know how to read. That you don't know how to read, but once you have this particular one, you can clearly understand and you can go ahead with how to read this particular protocol. Just give me a second. I'm trying to find out one other protocol. I'm not able to get this. Just give me a second. Okay, I'll check and I'll share the protocol with you. So that protocol will be the original protocol that was used by one of the clinical trials that we can use it for the reference purpose. All right. So what we need to discuss after this, we know what is protocol. We know who are going to prepare it, with the help of whom it will get prepared. What are the elements that are involved as part of your protocol? Because it is the entry point for your clinical trials. So the next step is going to be, we need to understand each and every topic or each and every parameter which is part of your protocol document. So once we complete that, then we need to understand the cross-functional interaction. That is nothing but you have a sponsor, you have an investigator or clinician, or you have a statistician, you have a data manager, and you have a medical writer. We are going to work with all of them, being a clinical SAS programmer, right? So we need to understand the role of each of this particular individual. So what is the role of a sponsor? What is the role of an investigator or a clinician? What is going to be the role of your biostatistician? What is going to be the role of your clinical data management team? So what is going to be the role of your medical data? So when you go to the clinical data management team, you are going to understand something very interesting, which is called as the CRF, case report form. So how to how to understand this particular case CRF form. What is called ECRF? What is called ACRF? All these terms we are going to understand and we are going to clear, right? So we may need 2 more classes to understand all these definitions. So from tomorrow's class onwards, I'm going to explain about the definitions. So the definitions, I'm going to start with protocol. So we are going to understand all the details which are present in all the elements which are present in the protocol, and then we understand what are the various teams that are involved in the clinical trials and what is the role of each of this particular roles and responsibilities of this particular team. And then we are happy to enter into the most important the interesting part of your clinical trials or clinical SAS, which is nothing but the testing. Before we start this particular one, we need to understand this. So to get this particular base, we may take a little time, but it won't take too much of time. So ideally, 2 classes are more than sufficient. Is that clear, guys? Is that okay to everyone? Is that understandable? Anything else you would like to discuss here? All right. Okay. Now, so what are the things that are pending from my end? So to start with this particular one, or if you want to refer something, first thing I need to provide is I need to provide you the protocol template. And second thing, I need to send you one protocol example. That is, I need to send you the protocol template. I also need to send you the protocol example or sample. So these 2 I will send along with this class notes. So you don't need to worry about the class notes. I will share the entire class notes to you. Even the clinical trials, phase 1, phase 2, phase 3, phase 4 is there, right? That one also I'm going to prepare it and I'm going to send it to you because it is there here. Here you can see, but I will put it in a proper format and I'll send it across to you. Clear? Mean to say, send it across to you means I will give it to Galusa. He will upload it in the questions, anyone? All right. So I'm going to wrap up the session here. Right? It's already 55 minutes we have started the session, and it went well, I believe. So any questions, I'm happy to answer that. We can have a Q&A now. If you don't have any question, we can wrap up the session for today. So perfect, everyone. See you tomorrow. Take care.